What if we could actually rebuild parts of the human brain? For decades, that's been total science fiction, right? Well, a really groundbreaking new clinical trial for Parkinson's disease, just published in the journal Cell, might be turning that fiction into a reality. And today, we're going to break down exactly how they're doing it. So, here's the big question, the one that scientists have been chasing for years. Can we really replace the specific brain cells that are lost in Parkinson's? For so long, treatments have just been about managing the symptoms. But this new approach? It's all about going right to the source of the problem. So, let's start with the challenge itself. To really appreciate just how incredible this new solution is, we've got to understand the problem. What is actually happening inside the brain of someone with Parkinson's disease? Okay, so at its core, Parkinson's is all about a chemical called dopamine. Dopamine is a neurotransmitter. Think of it like a critical messenger that helps our brain control movement, motivation, you name it. In Parkinson's, the very specific brain cells that make this chemical start to die off. And that loss is what leads to all the motor symptoms the disease is known for. A really great way to picture this is to imagine these cells as tiny little dopamine factories. They're all clustered in one specific part of the brain, and their whole job is to pump out dopamine to keep our movements smooth and controlled. But in Parkinson's, these little factories start shutting down one by one. So if the problem is failing dopamine factories, the idea behind the study is, well, it's pretty elegant, really. What if we could just build brand new ones from scratch and put them back in the brain? And the starting ingredient for these new factories? Human embryonic stem cells. The reason these cells are so incredibly powerful is because they're what scientists call pluripotent. That's just a fancy way of saying they are the ultimate blank slate. They have the amazing potential to become literally any type of cell in the entire body. So, how do you get a blank slate to become a highly specialized dopamine factory? Well, you can think of it like a very, very sophisticated biological recipe. Researchers use this cocktail of chemicals to basically give the stem cells instructions, guiding them step by step. First, they kind of nudge them to become general brain cells, and then they give them one last final instruction to turn into the exact type of dopamine-producing cell that's destroyed by Parkinson's. It's amazing. Now, making these cells in a petri dish is one thing, a huge accomplishment for sure, but the real test, the moment of truth, is finding out if they can actually work inside a living human brain. And that brings us to this first-in-human clinical trial. This graphic right here gives you a fantastic bird's-eye view of the whole process. You can see it all laid out, starting with creating the cells in the lab, then the actual transplantation into the patients, and finally, how they measure the results, using both physical tests and some really advanced brain imaging. And at the absolute heart of this study are 12 people. 12 pioneers, really, volunteers with Parkinson's disease, who bravely signed up for a procedure that, until them, had never been done in a human before. To really test this out, the researchers split the 12 participants into two groups. The first group got what they called a low dose, about 3.15 million cells. The second group got a high dose, double that, at 6.3 million cells. And the reason they do this is to see, does more cells actually lead to a better result? Now, this is so important to understand about a phase 1 trial like this one. Of course, everyone is hoping for a cure, but the number one, top priority above everything else is safety. Researchers had to answer one simple question first. Can we do this without causing any harm? Okay, so the stage was set. The main goal was proving it was safe. But after a year, what did the evidence actually show? Let's get into it. And here is the first and most important result. The treatment passed the safety test with flying colors. I'm quoting directly from the paper here. No dose-limiting toxicities or graft-related adverse events were observed. What that means in plain English is that the new cells were well tolerated. They didn't cause any major, scary side effects related to the treatment. That is a huge win. So check one, it's safe. That's a massive milestone. But of course, the question on everybody's mind is, did it actually help the patients? Did their symptoms get any better? All right, here's the clinical data. This chart is tracking motor symptoms over 12 months. Now, the key thing to know when you're looking at this is that a lower score is better. It means a person's movement has improved. And just look at the results. After a year, both groups showed real improvement. And what's even more exciting, the high-dose group, that's the one in blue, saw the biggest drop in their scores.
But here's the million-dollar question. Was this improvement really coming from those new cells? Well, these PET scans of the brain give us the answer. These scans are designed to literally light up areas where there's dopamine activity. Now, compare the top row, that's the brains at the start, to the bottom row, which is after 12 months. Do you see those brighter, hotter colors popping up? That is visible biological proof that the new dopamine factories have come online and are doing their job.
And this is the part that gets scientists so excited. There was this powerful connection. The patients who showed the most improvement in their movement tests were the same patients whose brain scans showed the biggest increase in dopamine activity. So the feeling of getting better was backed up by cold, hard biological proof. These results are honestly incredible. But with any pioneering science, it's really important we put them in context and look at what this all means for the future. And the researchers themselves are very upfront about the study's limitations. Look, it was a very small group, only 12 patients, and there wasn't a placebo group to compare against. Plus, we're only looking at the first year of data. So while this is a phenomenal start, we absolutely need larger, longer-term, and controlled trials to be sure about these benefits. So what's the big takeaway here? The author's own conclusion really sums it up perfectly. They say that transplanting these lab-grown cells is safe, and it shows the potential to improve the motor symptoms of Parkinson's. It's that classic, cautious, but optimistic language you see in good science, balancing the excitement with the need for more proof. When you step back, this one study is a monumental proof of concept. It's the first real hint that repairing the human brain might actually be possible one day. And the profound question it leaves us with is this: are we witnessing the dawn of a whole new era for treating disorders of the brain?